So, Hershey Chase grew viruses and cultures containing radioactive isotopes of phosphorus and sulfur. Okay, so radioactive isotopes basically just act as markers. So, if you have something radioactive, then you can sort of follow it. It's kind of like GPS for material, like before GPS. Like it's like having like a one of those air tags, right? That they have for the iPhones, where you can like put on your luggage or your purse or whatever. So that's what having a radioactive isotope is. It's basically a tag that you can then follow quite easily. Um, and the reason why they chose phosphorus 32 and sulfur 35 is because phosphorus 32 would help to tag DNA, because DNA doesn't have any sulfur. And the reason why they chose sulfur 35 is because the sulfur 35 would help tag the protein. Because again, they're trying to figure out which molecule is responsible for carrying genetic information of the virus. And again, their choices are it's either the DNA inside the virus, or it is the protein coat of the virus. So they need to tag both of them to figure it out. What they end up finding is that if the sulfur 35 is in the bacteria, then that means the virus protein had been injected in the bacteria. If they find the P32, then they know that the DNA core has been injected. So again, the sulfur 35 and the phosphorus 32 are just acting as what are they called, as like tags, so that they can then follow them. Um so what do they end up doing? They mix the marked viruses with bacterial cell, and then they just wait a few minutes to see what the virus is going to inject, and then they test the bacteria for radioactivity. Okay, so that's what they end up doing. um and the picture on your screen right now, and I believe you probably have a similar picture in your notes, I hope, is pretty much the experiment that they did. The bacteriophage ended up injecting the bacteria, which we know, and then they ended up having to test it out. What they found was that, oops, I went too far. Pretty much nearly all of the radioactivity in the bacteria was from the phosphorus, the marker found in DNA. So, what this means, of course, is that they were able to conclude that the genetic material of the bacteriophage was DNA and not protein. Okay? That being said, let's take a quick look at the video and then we'll move on.
Turning the clock back to the 1940s and 50s, the scientific community was in the midst of a debate about the nature of the hereditary material that is responsible for passing our traits on to our children, to our children's children, and so on for generations. Pretty existential question, right? An experiment by Avery, MacLeod, and McCarty in 1944 first suggested that proteins were not responsible for heredity, as previously thought, and instead DNA was the culprit. However, given that their results flew in the face of the current thinking, not everyone was convinced. In science, a great deal of evidence is needed before a new scientific idea is accepted as fact. Therefore, in 1952, Alfred Hershey and Martha Chase carried out experiments which addressed a similar question. Was DNA the hereditary material, or was it protein, or was it something else? To solve this important puzzle, Hershey and Chase needed a simple model organism to solve this complex question, and a clever experiment. They chose the simplest organism possible, a bacteriophage, which is a virus that infects bacteria. This virus looks a bit like a lunar landing module and attaches to the outside of the bacteria. It then injects a substance into the bacteria to create many new copies of the virus. These newly manufactured viruses are released when the bacteria bursts. So the substance injected inside of the bacteria must contain the instruction manual for making new virus. It contains the hereditary material. But what was this material that the virus injected into its host? The answer to their question might shed light on the nature of heredity for all life on Earth. Viruses consist of two things: a DNA core surrounded by a protein coat. Hershey and Chase needed to figure out which of the two was injected into the bacteria. They ran 2 experiments, and in each they labeled one of the components. For this labeling, they used radioactive tags: radioactive sulfur for the protein and radioactive phosphate for the DNA. This was an elegant method because phosphate is only found in DNA and not in the amino acids that make up proteins, and sulfur is only found in proteins and not DNA. After the viruses infected the bacteria, the researchers examined whether the radioactive signal was detected inside the bacteria or left outside. To do so, they needed to separate the bacteria from the viruses attached to their outsides, and did so using a Warren blender, similar to that used for making milkshakes. Once separated, they then centrifuged the sample. Since bacteria are bigger, they were centrifuged to the bottom, while the smaller viruses remained at the top of the tube. When they prepared virus with sulfur-labeled protein and mixed it with bacteria, they found that most of the radioactivity remained with the virus and did not enter the bacteria. When they allowed the virus to replicate and collected the new viruses, less than 1% of the protein radioactivity from the viral parents were found in the newly replicated virus. So the protein was not being inherited. On the other hand, when they performed the same experiment using virus with its DNA labeled with phosphate, they found that most of the radioactive DNA was injected into the bacteria from the attacking virus, and much of this labeled DNA was inherited and passed on to the new virus generation. Hershey and Chase added to the mounting evidence, pointing the finger at DNA as the hereditary material. But they were aware that their experiments left a few unanswered questions, as experiments often do, and they were cautious in their conclusions. From their observations, they stated that DNA had some function in the multiplication of the virus, whereas proteins did not. Word about the Hershey-Chase experiment spread quickly and helped to convince many scientists that DNA indeed was the molecule of heredity, and the remaining skeptics could not hold out for much longer. A year later, Watson and Crick published their model of the DNA double helix, which provided an elegant explanation for how DNA with such a simple structure could be replicated and pass along information from parents to offspring. The Hershey-Chase experiment is considered a classic in biology because it tackled an important question with the use of a well-chosen model organism and an elegant experimental design in order to clearly distinguish between two competing hypotheses.
All right. So,